Hello and welcome back to the Forgotten Deep. Today, let's relax and wind down, in a place where curiosity is allowed and sleeping is preferred. Now let us begin. When scientists found life in boiling ocean water for most of human history, the ocean depths remained as mysterious and inaccessible as the surface of another planet. We knew more about the moon than we did about the crushing darkness that lies beneath 3 miles of seawater. We thought we understood the fundamental rules of life on Earth, that all living things depended, ultimately, on the sun. That photosynthesis was the foundation of every ecosystem. That certain extremes of temperature, pressure, and chemistry were simply incompatible with life. We were wrong. In 1977, a small crew of scientists descended into the perpetual night of the deep Pacific Ocean, expecting to find barren volcanic rock. Instead, they discovered thriving ecosystems clustered around superheated vents, where water emerged at temperatures that would melt lead. They found towering worms with no mouths or digestive systems, clams the size of dinner plates, and bacteria that didn't just survive in conditions we thought were lethal, they thrived in them. This is the story of one of the most profound scientific discoveries of the 20th century, a discovery that revolutionized our understanding of life's boundaries, rewrote the rules of biology, and fundamentally changed our approach to searching for life beyond Earth. This is the story of when scientists found life in boiling ocean water. Part 1. The Deep Unknown. Chapter 1. The Abyss. Before discovery. To understand the magnitude of the 1977 discovery, we must first understand what came before it. For centuries, the ocean depths represented the ultimate frontier, more remote, more hostile, and less accessible than any terrestrial environment. The ancient Greeks believed the ocean had no bottom, that it descended infinitely into an aquatic underworld. Even as late as the 19th century, the prevailing scientific view was that the deep ocean was essentially a lifeless desert. The reasoning seemed sound. No sunlight penetrated beyond a few hundred feet, temperatures approached freezing, pressure increased by 1 atmosphere for every 33 feet of depth, and food would be scarce to non-existent. In 1858, the British naturalist Edward Forbes proposed his azoic theory, arguing that no life could exist below 1,800 feet. This theory dominated scientific thinking for decades. Forbes reasoned that the combined stresses of pressure, cold, and darkness created conditions fundamentally incompatible with biological processes. But Forbes was wrong, though it would take many years to prove it. The first hints came in the 1860s, when telegraph cables laid across the Atlantic Ocean floor were retrieved for repair. Attached to these cables, scientists found living organisms, sponges, corals, and other creatures that had somehow colonized the deep seafloor. The discovery prompted skepticism and debate. Perhaps, some argued, these creatures had attached to the cables while they were near the surface and simply survived the descent. The debate intensified in 1872 when the HMS Challenger embarked on a 3.5-year scientific expedition that would revolutionize oceanography. Over the course of their journey, the Challenger crew took depth soundings, temperature measurements, and biological samples from the deep ocean. They dredged the seafloor and discovered hundreds of new species living at depths Forbes had declared impossible. The Challenger expedition established beyond doubt that life existed in the deep ocean. But the organisms they found were sparse, slow-growing, and seemed to eke out a marginal existence in the dark. They were scavengers and predators, living off the slow rain of organic matter drifting down from the sunlit surface waters above, what oceanographers would later call marine snow. This established the paradigm that would dominate biological oceanography for the next century. The deep ocean was habitable, but barely. It was a frontier region where life persisted at the edge of possibility, sustained entirely by the distant sun shining on the ocean's surface thousands of feet above. Every food chain, every ecosystem, every living thing ultimately traced its energy back to photosynthesis in the sunlit upper ocean. This paradigm seemed unassailable. After all, it reflected a fundamental law of biology. Energy flows through ecosystems, and the ultimate source of that energy on Earth is the sun. Plants and photosynthetic bacteria capture solar energy and convert it into chemical energy stored in organic molecules. Herbivores eat the plants, carnivores eat the herbivores, and decomposers break down the dead. But it all starts with sunlight. Or so we thought. Chapter 2. The rise of deep sea exploration. The 20th century brought new technologies that would finally allow scientists to explore the deep ocean directly. But these advances came slowly, limited by the tremendous engineering challenges of operating in an environment more hostile than outer space. In 1930, William Beebe and Otis Barton made the first deep-sea descent in a submersible vessel they called the bathysphere, a steel ball 4.5 feet in diameter with tiny quartz windows. Suspended from a cable, they descended 1,426 feet into the ocean off Bermuda. Beebe described looking out into a world of utter darkness, punctuated by the bioluminescent flashes of deep-sea creatures. The bathysphere represented a triumph of human ingenuity, but it was also profoundly limited. Tethered to the surface by a cable, it could not maneuver or explore. It could only descend, observe, and ascend. Still, Beebe's vivid accounts of his descents captured public imagination and demonstrated that humans could, with proper equipment, venture into the deep. The next major advance came in the 1960s with the development of free-swimming submersibles, small submarines designed specifically for deep ocean research. The most famous of these was the Alvin, a deep submergence vehicle commissioned by the U.S. Navy in 1964 and operated by the Woods Hole Oceanographic Institution. Alvin was a remarkable machine. Built from steel and titanium, equipped with powerful lights and mechanical arms, it could carry a crew of 3, a pilot and 2 scientific observers, to depths of 6,000 feet, later upgraded to nearly 15,000 feet. Unlike the bathysphere, Alvin could move freely through the water, navigate complex terrain, and collect samples from the seafloor. But despite these technological advances, the paradigm of the deep ocean remained unchanged. The depths were cold, dark, and sparsely populated. Life existed there, yes, but it was entirely dependent on the productivity of the sunlit surface waters. The deep ocean was, in essence, a vast desert with occasional oases where currents concentrated the meager rain of organic debris from above. At the same time Alvin was being developed, geologists were making their own revolution in understanding the ocean floor. In the 1960s, the theory of plate tectonics unified decades of geological observations into a single, powerful framework. The Earth's surface, scientists realized, consisted of massive plates that moved slowly over geological time, driven by convection currents in the semi-molten mantle below. Where plates pulled apart, new oceanic crust formed at mid-ocean ridges, vast underwater mountain ranges that snake through all the world's ocean basins for over 40,000 miles. Here, magma welled up from the mantle, creating new seafloor and pushing the existing crust aside at rates of a few inches per year. This was dramatic geology, but its biological implications seemed limited. The mid-ocean ridges were remote, deep, and hostile. They were regions of active volcanism, earthquake activity, and newly formed rock. Surely, they were among the least likely places to find life in the deep ocean. Surely. Chapter 3. The plate tectonics revolution. The acceptance of plate tectonics in the late 1960s transformed geology from a largely descriptive science into a predictive one. For the first time, scientists could explain earthquakes, volcanoes, mountain formation, and the distribution of fossils within a single theoretical framework. But the theory also made predictions that could be tested. One prediction concerned the mid-ocean ridges. If new oceanic crust was continuously forming at these underwater mountain ranges, then the ridges should be geologically young, volcanically active, and hot. The rocks should still retain heat from their formation, and this heat should be slowly dissipating into the overlying ocean water. In the early 1970s, oceanographers began to study the thermal properties of mid-ocean ridges more carefully. They towed temperature sensors across the ridges and measured the heat flow through the seafloor. The results were puzzling. In some areas, heat flow measurements matched predictions. The rocks were warmer than the surrounding seafloor, as expected. But in other areas, the measurements were far too low. It was as if heat was somehow being removed from the ridge faster than the models predicted. One scientist pondered this mystery with particular intensity. John Edmund, a geochemist at MIT, realized that water circulating through cracks and pores in the hot rock could carry away tremendous amounts of heat through convection. Cold seawater would seep down through the fractured volcanic rock, become heated by contact with the hot underlying magma, and then rise back up to the surface. This hydrothermal circulation, Edmund reasoned, could explain the unexpectedly low heat flow measurements. But if this was happening, where was the hot water coming out? It had to be venting somewhere along the ridge, and when it did, it should carry with it dissolved minerals leached from the rock. The venting hot water might create unusual chemical signatures in the seawater or even deposit minerals on the seafloor. It was an elegant theory, but proving it would require going to the mid-ocean ridge and looking directly. And in 1977, that's exactly what a team of scientists proposed to do. Chapter 4, Project Famous and ITS Legacy. In 1974, 3 years before the transformative discovery, an international team of scientists conducted a pioneering expedition called Project Famous, the French-American mid-ocean undersea study. Using Alvin and two French submersibles, the team made dozens of dives to the mid-Atlantic ridge southwest of the Azores. Project Famous was primarily a geological expedition. Scientists wanted to observe the creation of new oceanic crust firsthand, to study the volcanic processes at work, and to test the predictions of plate tectonics theory. Over the course of the expedition, they made detailed observations of the ridge topography, collected rock samples, and documented active volcanic features. What they did not find was much in the way of life. The ridge was largely barren, as expected. Occasional fish swam past the submersible's lights, and sparse populations of typical deep-sea organisms were observed. But there was nothing unusual, nothing that challenged the prevailing understanding of deep ocean ecosystems. However, Project Famous did accomplish something important. It demonstrated that systematic exploration of mid-ocean ridges using submersibles was feasible. The technology worked, the techniques were sound, and the scientific value was clear. The expedition established protocols and procedures that future expeditions would follow. It also piqued scientific curiosity about other mid-ocean ridges. The Atlantic Ridge studied in Project Famous was a slow-spreading ridge, where the tectonic plates moved apart at rates of only an inch or two per year. But in the Pacific Ocean, there were fast-spreading ridges where plates separated at rates up to 6 inches per year. Would these faster-spreading ridges show different geology, different volcanic activity, different heat flow patterns? In 1976, a team of scientists proposed an expedition to the Galapagos Rift, a mid-ocean ridge in the eastern Pacific Ocean south of the Galapagos Islands. This was a fast-spreading ridge, and geologists expected it to show vigorous hydrothermal activity. The team included geologists, geochemists, and geophysicists. Their goal was to find and study hydrothermal vents, the places where hot water emerged from the seafloor. What they had no expectation of finding was life. At most, they thought they might observe some of the usual sparse deep-sea fauna. The idea that they would discover thriving ecosystems never entered their planning. They did not include a biologist on the initial expedition team. This oversight would become one of the expedition's most famous ironies. Chapter 5. Preparing for the Galapagos. The 1977 Galapagos expedition was meticulously planned. The team would use Alvin to explore a section of the Galapagos Rift at depths around 8,000 feet. Before the submersible dives, they would conduct a detailed survey of the seafloor using a towed camera sled called Angus, the acoustically navigated geological underwater survey system. Angus was essentially a heavy metal frame equipped with cameras, strobe lights, and temperature sensors. It would be towed just above the seafloor, taking photographs every few seconds while instruments recorded depth and water temperature. By analyzing the photographs and temperature data afterward, the team could identify promising locations for Alvin to investigate. In February 1977, the research vessel Nor arrived at the study site, and the team deployed Angus for a series of survey runs along the ridge axis. This was painstaking work. Each run lasted many hours. during which Angus photographed mile after mile of seafloor, while the team aboard nor monitored its depth and position. When Angus was recovered and the film was developed, the team gathered to examine thousands of photographs, looking for anything unusual. Most images showed the expected terrain: fresh volcanic rock, occasional pillow lava formations, sparse sediment cover. But then, in one frame, something unexpected appeared. The photograph showed what looked like white clam shells scattered on dark volcanic rock. This was odd. Clams in the deep ocean were not unheard of, but they were typically small and rare. These appeared to be unusually large and numerous. A few frames later, more strange objects appeared. The scientists could not immediately identify what they were seeing. Some images seemed to show whitish bacterial mats coating the rocks. Others showed what might be worm tubes or some kind of growth. The temperature data added to the intrigue. In the areas where these unusual organisms appeared, the sensors had recorded slight but measurable increases in water temperature, anomalies of a few tenths of a degree. It wasn't much, but in the deep ocean, where temperatures are stable within a fraction of a degree, these variations were significant. The team decided their first Alvin dive should investigate these anomalies. If nothing else, the temperature variations suggested they might be seeing evidence of hydrothermal venting, which was, after all, the expedition's primary objective. No one expected what they were about to find. Part 2. The Discovery Chapter 6. Dive 1. February 17th, 1977. On the morning of February 17th, 1977, pilot Jack Donnelly and observers Jack Corliss and Jerry Van Andel entered Alvin's pressure sphere. The hatch was sealed. The submersible was lifted over the side of the Nor and lowered into the Pacific Ocean. As the tether was released, Alvin began its descent into the abyss. The descent took over an hour. Sunlight faded within the first few hundred feet, giving way to twilight, then utter darkness. The only illumination came from the occasional flash of a bioluminescent jellyfish or squid drifting past. The pressure built steadily, 1 atmosphere for every 33 feet. At the seafloor, the pressure would exceed 3,600 pounds per square inch. Inside the titanium sphere, the three men monitored their instruments and maintained communication with Nor on the surface. The temperature gauge showed the water outside dropping to just above freezing as they descended. This was normal. In the deep ocean, temperatures hovered just a few degrees above 0°C, stable year-round regardless of surface conditions. Finally, Alvin's sonar indicated they were approaching the bottom. Donnelly switched on the exterior lights, and the seafloor materialized below them, dark volcanic rock, some areas covered with a light dusting of sediment. He brought Alvin down gently, and the submersible settled onto the bottom at a depth of 8,200 feet. The scene outside the viewport appeared at first to match expectations. Fresh-looking basalt stretched in all directions, with occasional pillow lava formations indicating relatively recent volcanic activity. This was consistent with their location on the ridge axis, where new crust was actively forming. But as Donnelly maneuvered Alvin forward, following the navigation coordinates toward the area where Angus had recorded temperature anomalies, the landscape began to change. The rocks took on a different appearance, lighter in color, with a whitish coating in places. Strange objects appeared in the lights. What looked like large white clam shells, some open, some closed, scattered across the volcanic rock. Jack Corliss, a geologist, stared at them in astonishment. These weren't the small, sparse clams typical of the deep sea. These were massive, 8, 10, 12 inches across. And there were so many of them. Then they saw the bacterial mats. Thick, white bacterial growth covered the rocks in some areas, forming ghostly blankets over the basalt. Nothing in their training or previous experience had prepared them for this. Deep-sea rocks were supposed to be nearly barren, with perhaps a thin biofilm of bacteria eking out an existence on the sparse organic matter that drifted down from above. This was different. This was abundant. This was thriving. As Alvin continued forward, more bizarre sights emerged from the darkness. Clusters of what appeared to be white crabs scuttled across the rocks. Strange worm-like tubes rose from crevices, their tips crowned with brilliant red plumes. Fish moved through the lights, more than the observers had ever seen in any previous deep-sea dive. and then they saw the water. Rising from fissures in the rock, shimmering columns of water distorted the view beyond them. The water was warm. Alvin's temperature sensors confirmed it. Anomalies of 5, 10, even 15 degrees above ambient temperature. For the deep ocean, this was extraordinary. This was proof of hydrothermal venting. But the vents were not what captured the observer's attention. It was the life. The sheer density, diversity, and vigor of the organisms clustering around these vents defied everything they thought they knew about deep-sea ecology. Jerry Van Andel, staring at the scene, finally spoke what they were all thinking. This doesn't make any sense. Chapter 7. The Garden of Eden. Over the next several weeks, Alvin made dive after dive to the Galapagos Rift. Each descent revealed new wonders, new mysteries, and new organisms that seemed to violate the fundamental rules of biology as the scientists understood them. They discovered entire fields of giant tube worms, later named Riftia pachyptila. These extraordinary organisms grew up to 8 feet tall, their white tubes anchored in the volcanic rock, their crimson gill plumes extending into the water. Most remarkably, they had no mouth, no digestive tract, no obvious way to feed. They found clusters of giant white clams, Calyptogena magnifica, some over a foot long, far larger than any deep-sea clam previously known. These clams were not just surviving. They were growing at rates that seemed impossible for deep-sea organisms, which typically grew extremely slowly due to the scarcity of food. They observed swarms of unusual white crabs and shrimp congregating around the vents, seemingly untroubled by water temperatures that should have cooked them. They documented dense bacterial mats that coated the rocks in thick layers, not sparse biofilms, but robust communities. One vent site, which the team dubbed Rose Garden for the red plumes of the tube worms, harbored such an abundance of life that it reminded observers of a tropical reef ecosystem. But in total darkness, at crushing pressure, over 1.5 miles below the ocean surface. The scientists collected specimens of everything they could. Using Alvin's mechanical arms, they carefully extracted tube worms, clams, crabs, and rocks covered with bacterial mats. These samples were brought to the surface in insulated containers, though many of the organisms died during the ascent due to the rapid pressure and temperature changes. But even dead or dying, the specimens provided crucial material for analysis. Back on Nor, the team examined the organisms under microscopes, dissected them, and preserved tissue samples for detailed study back on land. What they found only deepened the mystery. The tube worms' bodies contained an unusual organ called a trophosome, packed with bacteria. The clam's gills similarly harbored dense bacterial populations. Under the microscope, these bacteria appeared unusual, their cells containing strange inclusions and membranes. Most puzzling was what the organisms lacked. The tube worms had no digestive system at all, no mouth, no stomach, no intestines. The clams had a digestive tract, but it appeared reduced and perhaps non-functional. How were these animals obtaining nutrition? The initial hypothesis was that they must be filter feeders, somehow capturing bacteria from the water and digesting them. But the numbers didn't add up. The bacterial concentration in the vent water, while higher than normal deep-sea water, was nowhere near high enough to support such large, dense populations of animals through filter feeding alone. There had to be another explanation. Chapter 8. The chemosynthesis hypothesis. Back on shore, biologists examining the samples from the Galapagos Rift struggled to make sense of what they were seeing. The ecosystem's productivity was astonishing. Biomass estimates suggested these vent communities were as productive per unit area as tropical rainforests or coral reefs, the most productive ecosystems on Earth's surface, all powered by abundant sunlight. But these vent ecosystems existed in total darkness. There was no photosynthesis. So where was the energy coming from? The answer, when it came, represented a paradigm shift in biology. A scientist named Colleen Cavanaugh, then a graduate student at Harvard, proposed a radical idea. What if the bacteria living inside these organisms weren't parasites or symbionts in the conventional sense, but rather primary producers? What if they were generating organic matter not through photosynthesis, but through chemosynthesis? Chemosynthesis was not a new concept. Scientists had known since the late 19th century that certain bacteria could obtain energy from chemical reactions, specifically from oxidizing reduced chemicals like hydrogen sulfide, methane, or ammonia. These bacteria use the energy from these chemical reactions to fix carbon dioxide into organic molecules, much as photosynthetic organisms do using sunlight. But chemosynthetic bacteria were thought to be rare, marginal organisms that played a minor role in most ecosystems. The idea that they could support entire ecosystems, that they could be as productive as photosynthetic organisms, seemed far-fetched. Yet the more the scientists examined the vent ecosystems, the more sense Kavanagh's hypothesis made. The hydrothermal vent water was rich in hydrogen sulfide and other reduced chemicals. These chemicals represented a concentrated energy source, far more abundant than the sparse organic matter drifting down from the surface. If bacteria could tap into this chemical energy and use it to produce organic matter, and if these bacteria lived inside the tissues of animals like tube worms and clams, then a new kind of symbiosis became possible. The bacteria would have a stable environment and access to both the chemicals they needed from the vent water and the oxygen dissolved in seawater. In return, they would produce organic compounds that the host animal could use as food. This would explain the lack of a digestive system in tube worms. They didn't need one. They weren't eating. Their nutrition came from the bacteria living inside them, bacteria that were, in essence, farming chemicals. The hypothesis was revolutionary, but it needed testing. Scientists began examining the bacteria more closely, looking for evidence of chemosynthetic metabolism. They measured the chemical composition of the tube worm's tissues, looking for signatures of sulfide oxidation. They studied the biochemistry of the symbiosis, trying to understand how the animal and bacteria communicated and exchanged materials. The evidence mounted. The bacteria had the cellular machinery for sulfide oxidation and carbon fixation. The animals had specialized adaptations for concentrating sulfide and delivering it to their bacterial symbionts. The chemical signatures were consistent with a chemosynthesis-based food web. By 1981, the chemosynthesis hypothesis had gained widespread acceptance. The vent ecosystems represented a fundamentally new type of biological community, the first ecosystems discovered on Earth that did not depend, either directly or indirectly, on photosynthesis. They were powered not by the sun, but by the heat and chemistry of the Earth itself. Chapter 9. A new paradigm. The discovery of chemosynthetic ecosystems at hydrothermal vents forced a re-evaluation of some of biology's most fundamental assumptions. For decades, ecology textbooks had presented photosynthesis as the universal foundation of life on Earth. Yes, there were decomposers and chemosynthetic bacteria, but these were portrayed as marginal players, cleaning up or recycling energy that ultimately came from the sun. The vent ecosystems proved this view was incomplete. Here was a thriving, productive ecosystem whose primary energy source was geochemical, not solar. The implications rippled through multiple fields of science. In ecology, it meant that energy flow through ecosystems could follow pathways that didn't involve sunlight. The classical food chain, sunlight to plants to herbivores to carnivores to decomposers, had an alternative. Chemical energy to chemosynthetic bacteria to symbiont hosts to predators. In evolution, it raised new questions about the origins of the vent fauna. The tube worms, clams, and other vent organisms were clearly specialized for this environment. But how long had they been evolving there? Had vents been supporting similar ecosystems for millions of years? How isolated were these communities from the rest of the deep ocean? Genetic analysis of vent organisms provided some surprising answers. The tube worms, for instance, were related to other polychaete worms, but represented a highly divergent lineage. The giant clams belonged to a family of clams found in cold seeps, areas where methane and other reduced chemicals seep from sediments. This suggested that chemosynthetic symbioses had evolved multiple times in different lineages, adapting to different chemical environments. In biogeography, the discovery prompted expeditions to other mid-ocean ridges. If hydrothermal vents existed at the Galapagos Rift, they should exist at other ridges as well. Were they common? Did they support similar ecosystems worldwide? Over the following years, scientists discovered hydrothermal vents at ridges throughout the Pacific, Atlantic, and Indian Oceans. Each ridge system had its own vent fauna, with some species found globally and others endemic to specific regions. The global vent system represented a vast, interconnected network of ecosystems, most unknown to science until the late 1970s and early 1980s. Perhaps most profoundly, the discovery changed how scientists thought about the requirements for life. If organisms could thrive in the crushing pressure, total darkness, and chemical extremes of hydrothermal vents, then clearly life was more adaptable, more resourceful, and more persistent than anyone had imagined. This realization would have profound implications for astrobiology, the search for life beyond Earth. Chapter 10. The Smoking Black Plumes in 1979. Just 2 years after the initial discovery at the Galapagos Rift, scientists returned to the mid-ocean ridge, this time to the East Pacific Rise, a fast-spreading ridge system. Using Alvin, they explored deeper sections of the ridge, and what they found astonished even those who had seen the Galapagos vents. At depths approaching 9,000 feet, the team discovered a new type of vent. Unlike the warm, diffuse flows at the Galapagos, these vents erupted as vigorous jets of superheated water, black with suspended minerals. The temperature sensors maxed out. The water was hotter than 350 degrees Celsius, over 650 degrees Fahrenheit. These black smokers, as they came to be called, were far more dramatic than anything observed at the Galapagos. The superheated water erupted from chimney-like structures built from deposited minerals, sometimes rising tens of feet above the seafloor. The water was so hot that it should have been boiling, but the immense pressure of the deep ocean kept it in liquid form. As the superhot vent water mixed with the near-freezing seawater, dissolved minerals precipitated out in billowing black clouds, giving the vents their name. Over time, these minerals accumulated, building the chimney structures and depositing rich or bodies on the seafloor, natural massive sulfide deposits of the type mined on land. The chemistry of black smoker fluids was extreme. The water was highly acidic, with pH values as low as 2 or 3, as acidic as vinegar. It contained high concentrations of metals like iron, copper, zinc, and lead, along with toxic levels of hydrogen sulfide. This was an environment that seemed wholly incompatible with life. Yet life was there. Around the bases of the black smokers, similar communities of tube worms, clams, and crabs congregated. Even more remarkably, scientists discovered bacteria living directly in the vent fluids, thriving at temperatures that would denature proteins in most organisms. These thermophilic bacteria represented an even more extreme form of life than the vent animals. They had adapted to survive and reproduce at temperatures that would sterilize medical equipment. Their enzymes remained stable and functional under conditions that would destroy the cellular machinery of most life on Earth. The discovery of black smokers and their associated hyperthermophilic bacteria pushed the known boundaries of life even further. Not only could life exist without sunlight, it could exist in chemical and thermal conditions previously thought to be fundamentally lethal. Scientists began to study these extremophiles, organisms that thrive in extreme conditions, with intense interest. What adaptations allowed them to survive? How did their cellular machinery function under such stress? And most intriguingly, what could they tell us about the origins and limits of life? Part 3. Understanding the Impossible. Chapter 11. The biology of extremophiles. The organisms living at hydrothermal vents challenged biologists to rethink what was possible. To survive and thrive in such extreme conditions required fundamental adaptations at every level of biological organization, from the structure of individual molecules to the architecture of entire ecosystems. Consider the problem of temperature. For most organisms, proteins, the molecular machines that carry out cellular functions, have an optimal operating temperature range. Too cold, and they become sluggish and inefficient. Too hot, and they begin to unfold and lose their three-dimensional structure, a process called denaturation. For most life on Earth, proteins denature at temperatures above about 40 to 50 degrees Celsius. Yet the bacteria living in black smoker fluids were thriving at temperatures above 120 degrees Celsius. How? The answer lies in the structure of their proteins. Thermophilic bacteria produce proteins with modifications that stabilize them at high temperatures. These modifications include increased numbers of ionic bonds, additional disulfide bridges, and a more compact overall structure. The proteins of hyperthermophiles are naturally more rigid than those of mesophilic organisms, organisms that prefer moderate temperatures. This rigidity is a trade-off. At moderate temperatures, thermophile proteins would be too stiff to function properly. But at high temperatures, when normal proteins would unfold and collapse, thermophile proteins retain their structure and continue to function. Beyond proteins, these organisms had to solve the problem of membrane stability. Cell membranes are made largely of lipids, fats, that form a bilayer around the cell. At high temperatures, normal lipids become too fluid, allowing the membrane to lose integrity. Thermophiles produce specialized lipids with longer chains and more cross-linking, creating membranes that remain stable even as temperatures soar. Even their DNA required special adaptations. DNA exists as a double helix, with the two strands held together by hydrogen bonds. High temperatures can cause these bonds to break, denaturing the DNA and making it inaccessible for reading and copying. Hyperthermophiles have evolved mechanisms to protect their DNA, including specialized proteins that bind to and stabilize the double helix, and the use of unusual bases that form stronger bonds. But temperature was just one of many challenges these organisms faced. The pressure at 8,000 or 9,000 feet below the surface is immense, over 3,600 pounds per square inch, or about 250 atmospheres. This is enough to compress gases and affect the structure of proteins. Yet pressure turned out to be less of a barrier than scientists initially expected. Unlike gases, liquids and solids are largely incompressible. The water inside a cell at high pressure is under the same pressure as the water outside, so there's no net force trying to crush the cell. Proteins do experience some structural effects from pressure, but many organisms have adapted surprisingly well. The chemistry posed perhaps the greatest challenge. Hydrothermal vent water contains compounds that are toxic to most life. Hydrogen sulfide, the compound that gives rotten eggs their characteristic smell, is present at concentrations that would kill most animals within minutes. It interferes with cellular respiration by blocking the electron transport chain in mitochondria. The vent animals have evolved sophisticated mechanisms to handle sulfide. The giant tube worm Riftia pachyptila produces specialized hemoglobin molecules that can bind both oxygen and sulfide simultaneously, transporting both to the bacteria in its trophosome. The hemoglobin prevents free sulfide from reaching tissues where it would be toxic, while delivering it to the bacteria that need it for energy. The tube worm's bright red color comes from this hemoglobin. Unlike human hemoglobin, which contains iron, the tube worm's hemoglobin has additional binding sites that allow it to safely sequester sulfide. It's an elegant solution to a deadly problem. Chapter 12. Symbiosis at the extreme. Perhaps the most remarkable adaptation in vent ecosystems is the symbiosis between animals and chemosynthetic bacteria. This relationship represents one of the most intimate forms of cooperation in nature, a partnership that allows both parties to exploit an energy source unavailable to either alone. Consider again the giant tube worm Riftia pachyptila. This animal has no mouth, no gut, and no obvious way to obtain food. Instead, nearly one-third of its body weight consists of bacteria living in a specialized organ called the trophosome. How did this extraordinary symbiosis evolve, and how does it work? The bacteria living in the trophosome are chemosynthetic sulfide oxidizers. They obtain energy by converting hydrogen sulfide and oxygen into sulfate, and they use this energy to fix carbon dioxide into organic molecules, sugars, amino acids, and other compounds that can be used as food. The tube worm provides the bacteria with everything they need: a stable environment, protection from predators, and delivery of both sulfide and oxygen. The worm's circulatory system brings oxygenated blood from the plume, where it exchanges gases with seawater, and sulfide-rich blood from the base of the tube, where it is exposed to vent water. These are transported to the trophosome, where the bacteria use them. In return, the bacteria share the organic compounds they produce with their host. The tube worm absorbs these compounds and uses them for growth, reproduction, and energy. It's a complete nutritional relationship. The worm depends entirely on its bacterial symbionts for food. This symbiosis allows Riftia to grow remarkably fast for a deep-sea organism. Studies have measured growth rates of over 30 inches per year in some tube worms, extraordinarily rapid for an animal living in the deep ocean, where most organisms grow slowly due to limited food. This fast growth is possible because the worm has essentially tapped into a rich energy source and automated the process of capturing it. The giant clams of vent ecosystems have a similar but distinct symbiosis. Unlike tube worms, clams do have a digestive system, but it appears to be largely non-functional. Instead, chemosynthetic bacteria live in the clam's gills, where they have access to both the vent water that the clam filters and the oxygen in seawater. The clam's gills are enormously enlarged compared to related shallow water clams, a modification that provides more space for the bacterial symbionts. The bacteria oxidize sulfide and produce organic matter, which the clam absorbs. The clam can also filter feed, capturing free-living bacteria from the water, but its primary nutrition comes from its symbionts. What makes these symbioses even more remarkable is their specificity. The bacteria living in Riftia are not found free-living in the environment. They exist only in association with the worms. Similarly, the bacteria in vent clams represent specific strains adapted to that particular symbiosis. This raises fascinating questions about how the symbiosis is maintained across generations. Tube worms are not born with their bacterial partners. Instead, each larval worm must acquire the bacteria from the environment. Yet somehow, the worm selects and cultivates the correct bacterial species, while excluding the thousands of other bacterial species present in vent water. Scientists are still working to understand the mechanisms of this selectivity. The worm appears to produce chemical signals that attract the right bacteria, and its immune system recognizes and tolerates these symbionts while attacking potential pathogens. It's a complex molecular dialogue between partners, refined over millions of years of coevolution. The result is one of nature's most successful partnerships, a symbiosis that allows organisms to thrive in one of Earth's most extreme environments. Chapter 13. The vent food web. While the symbiotic relationships between animals and bacteria form the foundation of vent ecosystems, these systems support a complex web of other organisms as well. The vent communities are not isolated pairs of symbionts, but rather dynamic ecosystems with predators, scavengers, and competitors all playing their roles. Around the tube worm clusters and clam beds, scientists have documented an impressive diversity of life. There are specialized vent fish, including species of eelpouts and grenadiers that seem to favor the vent environment. These fish feed on the smaller crustaceans and worms that swarm around the vents. The white crabs observed in the first Galapagos dives, later identified as Bythagria thermidron, are among the most common vent animals. These crabs are omnivores, feeding on bacteria, detritus, and occasionally preying on smaller animals. They have been observed farming bacteria, scraping them off rocks, and consuming them. Dense populations of limpets graze on bacterial mats, and various species of polychaete worms burrow in the sediments around vents, likely feeding on bacteria and organic debris. Vent shrimp crowd around black smokers, some species positioning themselves directly in the plumes of hot water. One species of vent shrimp, Rimicaris exoculata, discovered at mid-Atlantic ridge vents, has particularly unusual adaptations. These shrimp lack functional eyes, but have developed specialized light-detecting organs on their backs. Why? Because black smoker vents emit a faint infrared glow due to their extreme temperature. The shrimp can detect this light and use it to navigate, positioning themselves close enough to the vents to farm bacteria, but not so close as to be cooked. Perhaps most remarkably, Rimicaris hosts its own bacterial symbionts, not internally like tube worms and clams, but externally, in enlarged gill chambers. These bacteria appear to be chemosynthetic, providing nutrition to the shrimp. So the shrimp are both farmers of free-living bacteria and hosts to symbiotic bacteria, a dual strategy for exploiting the chemical energy of vents. Even within a single vent field, conditions vary considerably. Some areas receive direct flows of hot, sulfide-rich water. Others receive more diffuse flows where hot vent water has mixed with ambient seawater. Still other areas receive no vent flow at all, but may still benefit from the elevated productivity of the vent community. Different organisms specialize for different niches within this gradient. The most thermophilic bacteria colonize the hottest areas, often forming thick mats directly at vent orifices. Tube worms typically establish in areas with moderate flow, where they can access both sulfide and oxygen efficiently. Clams often occur in slightly cooler areas, and predators and scavengers range across the entire vent field. This zonation pattern creates a complex spatial structure to the ecosystem. It's similar in principle to the zonation seen in intertidal environments, where different organisms occupy different positions along the gradient from constantly submerged to regularly exposed. But instead of a tidal gradient, vent organisms are distributed along a thermal and chemical gradient. The result is a patchy, dynamic ecosystem. Vents are ephemeral. They may flow vigorously for years or decades, then shut off as the underlying volcanic plumbing changes. New vents open as tectonic activity creates new fissures. Organisms must be able to colonize new vents and persist until their current vent fails. This creates a population structure similar to that seen in fire-adapted forests or other disturbance-driven ecosystems. Vent organisms produce large numbers of larvae that disperse through the deep ocean, searching for new vents. Most perish, but a few find newly active vents and establish new populations. Over time, a network of vent populations is maintained across the global mid-ocean ridge system. Scientists studying vent ecosystems have documented succession, the sequence of species that colonize a new vent over time. The first colonists are typically fast-growing bacteria, followed by small, quick-reproducing animals. Later, larger, slower-growing species like giant tube worms establish and may dominate the community for years. Understanding these dynamics requires long-term observations. Beginning in the 1990s, scientists established long-term monitoring sites at several vent fields, returning year after year to document changes. These studies have revealed the sometimes dramatic shifts that occur as vents wax and wane, as species come and go, and as the ecosystem responds to volcanic eruptions, earthquakes, and other disturbances. Chapter 14. The global distribution of vents following the initial discoveries at the Galapagos Rift and East Pacific Rise, scientists began systematically exploring other mid-ocean ridges. What they found confirmed that hydrothermal vents are a global phenomenon, found at all major ridge systems. In the Atlantic Ocean, vents were discovered along the Mid-Atlantic Ridge, including the famous Tag hydrothermal field and the Lucky Strike vent field. These vents supported ecosystems similar in structure to Pacific vents, but with different species. The Mid-Atlantic Ridge hosts the shrimp-dominated communities mentioned earlier, quite different from the tubeworm and clam communities of the Pacific. In the Indian Ocean, vents were found along the Central Indian Ridge and the Southwest Indian Ridge. Again, the basic ecosystem structure was similar, chemosynthetic bacteria supporting various animals, but the species were distinct. Even in the Arctic Ocean, beneath the ice, scientists discovered hydrothermal vents at the Gackel Ridge. These vents, located in one of the most remote and inaccessible regions of the planet, supported their own unique communities of organisms adapted to both the chemical extremes of vents and the year-round near-freezing temperatures of Arctic waters. As the catalog of known vent sites grew, biogeographers began to analyze the patterns of species distribution. Some species showed remarkably wide distributions. Certain bacteria were found at vents worldwide. Other species were endemic to particular ridge systems or even individual vent fields. The patterns revealed insights into how vent organisms disperse and evolve. Larvae of vent animals drift in deep ocean currents, potentially traveling long distances before settling. But barriers to dispersal exist: deep trenches, continental margins, and equatorial current systems can all restrict larval movement. Over evolutionary time, populations separated by such barriers can diverge, becoming distinct species. This process has created a global patchwork of vent biodiversity, with each major ridge system hosting its own assemblage of species, some shared with nearby ridges, others unique. Scientists have estimated that global vent ecosystems may harbor thousands of species, many still unknown to science. Each new expedition to unexplored ridge sections discovers new organisms. Some are variations on familiar themes, a new species of vent clam, another type of tube worm. Others are completely novel, unusual organisms with no close relatives at other vents. One of the most significant discoveries came in the late 1980s, when scientists explored vents in the back arc basins near Japan. Here, they found the hairy snail, Chrysomalin squamiferum, a gastropod with unusual iron sulfide scales covering its foot. This snail farms bacteria on its enlarged gill surface and has become an icon of vent biodiversity. Vents have also been discovered in unusual settings beyond mid-ocean ridges. At the Mariana Volcanic Arc, vents emit fluids enriched in CO2 and sulfur from the subducting tectonic plate, creating unique chemical conditions. At the Vaughan Dam vent field in the Caribbean, vents occur off-axis from the ridge in an unusual tectonic setting. Each new vent discovery adds to our understanding of the global vent system and the adaptations of vent life. But vents are not the only chemosynthetic ecosystems in the deep sea. Chapter 15: Cold Seeps and Whale Falls. As scientists explored the deep ocean more systematically in the 1980s and 1990s, they discovered that hydrothermal vents were not the only places where chemosynthetic ecosystems thrived. Two other major types of chemosynthetic habitats were found: cold seeps and whale falls. Cold seeps occur in areas where hydrocarbon-rich fluids, methane, oil, and other organic compounds, seep slowly from sediments. These seeps are common along continental margins, particularly in areas of active petroleum generation or near tectonic plate boundaries. Unlike hydrothermal vents, cold seeps emit fluids at ambient temperature or even slightly colder than surrounding seawater. The term cold is relative. These are not hot like vents. But the chemistry is similar in principle. Reduced chemicals, in this case methane and sulfide, provide energy for chemosynthetic bacteria. Cold seep communities often include clams and mussels with chemosynthetic symbionts, similar to vent clams but belonging to different species. Bacterial mats are common, and carbonate rocks often form where microbial methane oxidation alters the local chemistry. Some seeps also support tube worms, including species related to vent tube worms. One fascinating group of organisms found at cold seeps are the ice worms, polychaetes that live in methane hydrate deposits. Methane hydrates are ice-like solids formed when methane is trapped in a lattice of water molecules under high pressure and low temperature. These worms burrow through the hydrates, grazing on bacteria. Cold seeps can persist for centuries or millennia, much longer than typical hydrothermal vents. This stability allows slow-growing organisms to establish and persist. Some seep clams live for over 200 years, among the longest-lived invertebrates known. Whale falls represent a third type of chemosynthetic ecosystem, and perhaps the most unexpected. When a large whale dies and sinks to the deep seafloor, its carcass represents a massive input of organic matter, typically 40 to 60 tons of tissue and bone. This bonanza attracts scavengers. Hagfish, sleeper sharks, and crabs arrive within days to feast on the soft tissues. But the whale fall supports life long after the scavengers have stripped the flesh. The lipid-rich bones continue to release organic compounds for decades. Specialized bacteria colonize the bones, breaking down the fats and generating sulfide as a metabolic byproduct. This sulfide, in turn, supports chemosynthetic bacteria, which attract animals with chemosynthetic symbionts. Scientists have documented mussels, clams, snails, and bone-eating worms, ostacs, that colonize whale falls. These organisms are often closely related to vent and seep fauna, suggesting an evolutionary connection between these different chemosynthetic habitats. The stepping stone hypothesis proposes that whale falls, cold seeps, and hydrothermal vents are all connected in an evolutionary sense. Organisms adapted to one type of chemosynthetic environment may be pre-adapted to colonize others. Whale falls, being more widely distributed than vents, may serve as stepping stones allowing vent organisms to disperse across ocean basins. This hypothesis gains support from genetic studies showing that some vent organisms have close relatives at seeps and whale falls. The ecosystems, while distinct, form a connected network of chemosynthetic habitats spanning the global ocean. Together, hydrothermal vents, cold seeps, and whale falls reveal that chemosynthetic ecosystems are far more common and important than scientists realized before 1977. They represent a substantial component of deep-sea biodiversity and play significant roles in ocean chemistry and carbon cycling. Part 4. The Science of Extremophiles Chapter 16. Defining life's limits. The discovery of life at hydrothermal vents prompted scientists to ask fundamental questions. What are the limits of life? What conditions are truly incompatible with biological processes? And how do we define these limits? Before 1977, scientists assumed they had a reasonable understanding of life's boundaries. They knew that most organisms required temperatures between about 0 and 40 degrees Celsius, neutral pH, moderate pressure, and the absence of toxic chemicals. Extremophiles, organisms adapted to extreme conditions, were known, but considered rare oddities, not major players in global ecosystems. The vent discoveries changed this perspective. Suddenly, scientists realized that extremophiles were not rare exceptions, but potentially abundant and important. This prompted systematic efforts to catalog and study organisms from extreme environments. Researchers began collecting samples from hot springs, acid lakes, hypersaline pools, deep subsurface rocks, polar ice, and other extreme habitats. They cultured microorganisms from these samples, studied their physiology and biochemistry, and mapped the limits of their tolerance. The results were astonishing. Microorganisms were found thriving in virtually every extreme environment tested. Thermophiles living in boiling hot springs at temperatures up to 80 degrees Celsius. Hyperthermophiles growing optimally above 80 degrees Celsius, with some surviving temperatures above 120 degrees Celsius. The current record is 122 degrees Celsius for Methanopyrus canleri. Psychrophiles growing at temperatures below 15 degrees Celsius, with some reproducing at temperatures as low as minus 15 degrees Celsius in Arctic sea ice. Acidophiles thriving at pH values below 2, equivalent to battery acid. Alkalophiles growing at pH values above 11, comparable to ammonia solutions. Halophiles requiring salt concentrations of 15 to 30%, far higher than seawater. Barophiles, or piezophile, adapted to the immense pressures of the deep ocean, with some requiring high pressure to grow. Radioresistant organisms surviving radiation doses that would be lethal to humans many times over. Perhaps most remarkably, some organisms, called polyextremophiles, thrive in multiple extreme conditions simultaneously. The Archean Pacrophilus tortoise, for instance, grows optimally at 60 degrees Celsius and pH 0.7, hot and extremely acidic. These discoveries revealed that life's boundaries are far broader than previously thought. But they also raised a new question. Are there any true limits? Can life exist under any conditions? Or are there fundamental barriers beyond which biological processes cannot occur? Theoretical work suggests there are indeed limits. At very high temperatures, chemical bonds essential to life, particularly hydrogen bonds and certain covalent bonds, become unstable. Above about 150 degrees Celsius, even the most sophisticated adaptations may be insufficient to prevent critical biomolecules from degrading. Similarly, at very low temperatures, water freezes and chemical reactions slow to imperceptibly low rates. While some organisms can survive freezing in a dormant state, active metabolism and reproduction appear to require at least some liquid water. Extreme pH values pose chemical challenges. Very acidic conditions can protonate biomolecules and disrupt their structure, while very alkaline conditions can deprotonate them. Organisms have evolved remarkable mechanisms to maintain neutral pH inside their cells while living in extreme pH environments. But there are limits to these mechanisms. Radiation and high pressure also have theoretical limits, though organisms have proven more resilient than expected in both cases. The bacterium Deinococcus radiodurans can survive radiation doses over 1,000 times higher than would kill a human, thanks to sophisticated DNA repair mechanisms. Deep-sea organisms routinely function at pressures over 1,000 atmospheres. While research continues to map these limits, one lesson has become clear: life is extraordinarily adaptable. Given liquid water and an energy source, life finds a way. Chapter 17. Archea, the third domain of life. The study of extremophiles led to another revolutionary discovery: a fundamental reorganization of the tree of life itself. For most of the 20th century, biologists divided all life into two primary categories: prokaryotes, simple cells without nuclei, including bacteria, and eukaryotes, complex cells with nuclei, including animals, plants, fungi, and protists. This division seemed fundamental, representing two distinct architectural plans for cellular organization. But in the 1970s, a scientist named Carl Woese began studying the evolutionary relationships among organisms using a new technique. Instead of comparing anatomy or physiology, Woese compared the sequences of ribosomal RNA, a molecule found in all cells and essential for protein synthesis. By comparing ribosomal RNA sequences, Woese could estimate how closely related different organisms were. Closely related organisms would have similar sequences. Distantly related organisms would have accumulated more differences over evolutionary time. What Woese discovered shocked the scientific community. Among the prokaryotes, organisms without nuclei, there were two fundamentally distinct groups. One group included the familiar bacteria, E. coli, cyanobacteria, proteobacteria, and so on. But the other group was profoundly different at the molecular level. Woese called this second group archaea. They looked like bacteria under the microscope, small, simple cells without nuclei. But their ribosomal RNA sequences were as different from bacterial sequences as bacterial sequences were from eukaryotic sequences. In some ways, archaea were actually more similar to eukaryotes than to bacteria. Further research revealed that archaea differed from bacteria in fundamental ways. Their cell membranes were built from different lipids, their genetic machinery used different enzymes, and their metabolic pathways included unique chemical reactions. These were not just unusual bacteria, they were a distinct form of life. In 1990, Woese proposed a radical reorganization of the tree of life. Instead of two primary divisions, prokaryotes and eukaryotes, there were three domains: bacteria, archaea, and eukarya. This three-domain system is now universally accepted in biology. What does this have to do with extremophiles and hydrothermal vents? It turns out that many extremophiles are archaea. In fact, many of the most extreme extremophiles known are archaeal species. The hyperthermophiles living in black smoker fluids at temperatures above 100 degrees Celsius, many are archaea. The methanogens, organisms that produce methane, that thrive in oxygen-free environments, they're all archaea. The halophiles living in salt lakes, many are archaea. The connection between archaea and extreme environments led some scientists to propose that archaea might represent an ancient form of life, adapted to the harsh conditions of early Earth. This idea has become controversial and remains debated, but it highlights an important point. The discovery of extremophiles and the recognition of archaea as a distinct domain of life are intimately connected. The molecular tools developed to study extremophiles helped identify archaea, and the study of archaea helped scientists understand how life can adapt to extreme conditions. Together, these advances revolutionized our understanding of life's diversity and evolutionary history. Chapter 18. Biochemical Adaptations. To extremes. Understanding how extremophiles survive requires examining adaptations at the molecular level. The biochemistry of extremophiles reveals an astonishing array of solutions to the challenges posed by extreme environments. Consider the enzymes of thermophiles and hyperthermophiles. Enzymes are biological catalysts. proteins that speed up chemical reactions in cells. For most organisms, enzymes work best at moderate temperatures. Heating them causes the protein to unfold and lose activity, a process called denaturation. Thermophile enzymes maintain their structure and function at high temperatures through several modifications. First, they have increased numbers of ionic bonds and salt bridges, electrostatic interactions between charged amino acids. These bonds are weak individually, but collectively stabilize the protein structure. Second, they have more compact structures with better packed interiors. This reduces the number of cavities where the protein could unfold. Third, many have increased numbers of disulfide bonds, covalent links between cysteine amino acids. These bonds are strong and resist breaking even at high temperatures. Fourth, thermophile proteins often have different amino acid compositions, favoring amino acids that promote stability. For instance, proline, a rigid amino acid, is more common in thermophile proteins. The result is an enzyme that remains folded and functional at temperatures that would destroy normal proteins. Remarkably, at room temperature, these enzymes are often inactive or very slow. They're too rigid. They require heat to become flexible enough to function properly. This property has made thermophile enzymes extremely valuable in biotechnology. One famous example is Taq polymerase, an enzyme from the thermophilic bacterium Thermus aquaticus. This enzyme is used in PCR, polymerase chain reaction, a technique for copying DNA. Because Taq polymerase is stable at high temperatures, it can withstand the heating steps required for PCR, making the process efficient and reliable. Acidophiles and alkalophiles face different challenges. They must maintain neutral pH inside their cells despite living in extremely acidic or alkaline environments. They accomplish this through specialized membrane pumps that actively transport protons, H plus ions, in or out of the cell, maintaining an internal pH near neutral even when the external pH is extreme. Halophiles cope with high salt concentrations using one of two strategies. Some organisms exclude salt from their cells and produce organic compounds, compatible solutes, that balance the osmotic pressure. Others allow salt to enter their cells but adapt all their proteins to function in high salt conditions. These salt-in halophiles have proteins that actually require high salt concentrations to maintain their structure. Psychrophiles, cold-adapted organisms, have enzymes with modifications that keep them flexible at low temperatures. They use opposite strategies to thermophiles, more flexible structures, fewer stabilizing bonds, and amino acid compositions that favor flexibility over rigidity. Perhaps most remarkably, some organisms produce antifreeze proteins that prevent ice crystals from forming in their cells. These proteins bind to tiny ice crystals and prevent them from growing, allowing the organism to survive at temperatures below the normal freezing point of water. Radio-resistant organisms like Deinococcus radiodurans have evolved sophisticated DNA repair systems. Radiation damages DNA by breaking the double helix and causing mutations. Most organisms can repair some DNA damage, but Deinococcus can repair multiple simultaneous breaks. Its DNA can be shattered into hundreds of fragments, and the organism will accurately reassemble it. The mechanisms of this repair are still being studied, but they appear to involve redundant copies of the genome, specialized repair enzymes, and cellular structures that hold the DNA fragments in the correct positions during repair. Chapter 19. Energy metabolism in extremophiles. Beyond surviving extreme conditions, extremophiles must also obtain energy and carry out metabolism. The diversity of energy harvesting strategies in extremophiles rivals the diversity of their environmental adaptations. We've already discussed chemosynthesis, the process by which organisms obtain energy from chemical reactions rather than sunlight. But there are many different types of chemosynthesis, each exploiting different chemical substrates. Some chemosynthetic bacteria oxidize hydrogen sulfide, converting it to sulfate and capturing the energy released. This is the strategy used by the symbionts in tube worms and clams. The overall reaction is H2S plus 2O2 SO4 2. plus 2H++ energy. Other chemosynthetic bacteria oxidize methane, hydrogen gas, ammonia, or even iron or manganese. Each of these reactions releases energy that can be used to fix carbon dioxide into organic matter. Methanogenesis, the production of methane, is another unique energy metabolism found exclusively in archaea. Methanogens reduce carbon dioxide to methane using hydrogen gas as the electron donor. CO2 plus 4H2CH4 plus 2H2O plus energy. This reaction occurs in oxygen-free environments and is responsible for the methane found in swamps, rice paddies, the guts of animals, and some hydrothermal vents. Methanogens were among the first hyperthermophiles discovered, with some species growing optimally above 100 degrees Celsius. Some extremophiles use sulfur compounds in place of oxygen as the final electron acceptor in their metabolism. Instead of cellular respiration that produces water, they carry out sulfur respiration that produces hydrogen sulfide. This allows them to generate energy in environments with no oxygen. Iron and manganese metabolism represents another unusual strategy. Some bacteria can oxidize ferrous iron, Fe2+, to ferric iron, Fe3+, capturing the energy released. Others reduce ferric iron to ferrous iron during their metabolism. These iron-metabolizing bacteria play important roles in geochemical cycling and have been found in hydrothermal vent environments. Perhaps most remarkably, some organisms can derive energy from radioactive decay. In 2008, researchers discovered bacteria living in South African gold mines nearly 2 miles below the surface, in water that hadn't been in contact with the surface for millions of years. These bacteria appeared to be deriving energy from the radiolysis of water, the splitting of water molecules by radiation from radioactive uranium in the surrounding rock. The diversity of energy metabolisms in extremophiles reveals a fundamental principle: life is opportunistic. Wherever there is a source of chemical energy and appropriate conditions, organisms evolve to exploit it. The discovery of chemosynthetic ecosystems at hydrothermal vents was just the beginning of this realization. Chapter 20. Applications of extremophile research. The study of extremophiles is not just academically fascinating. It has led to numerous practical applications in biotechnology, industry, and medicine. We've already mentioned Taq polymerase, the heat-stable enzyme from a hot spring bacterium that revolutionized molecular biology. PCR, using Taq polymerase, is now a standard technique used in research, medicine, forensics, and many other fields. The development of Taq polymerase generated billions of dollars in commercial value and countless scientific advances. But Taq is just one example. Many other extremophile enzymes have found commercial applications. Cold-active enzymes from psychrophiles are used in laundry detergents, allowing them to work effectively at low wash temperatures, saving energy and protecting delicate fabrics. Alkaline proteases from alkalophiles are used in detergents to break down protein-based stains at high pH. Thermostable amylases from thermophiles are used in industrial starch processing, converting starch to sugars at high temperatures more efficiently than conventional enzymes. Extremophile enzymes are also valuable in industrial processes that involve harsh conditions. For instance, paper bleaching uses chlorine compounds at high pH, and enzymes that can function under these conditions have improved the efficiency of the process. In DNA sequencing, extremophile enzymes have enabled new technologies. The original DNA sequencing methods were slow and labor-intensive. Modern high-throughput sequencing relies on thermostable polymerases and other enzymes that can withstand repeated heating and cooling cycles or function for extended periods under controlled conditions. Extremophiles themselves, not just their enzymes, have found applications. Some thermophilic bacteria are used in bioremediation, breaking down pollutants in hot industrial wastewater. Acidophilic bacteria are used in biomining, extracting metals from low-grade ores by oxidizing metal sulfides. In astrobiology, extremophile research guides the search for life on other worlds. If life can thrive in Earth's most extreme environments, then perhaps similar life exists in the subsurface of Mars, the ice-covered oceans of Europa, or Enceladus, or the methane lakes of Titan. We'll explore these astrobiological implications in detail later. Medical applications are also emerging. Some extremophiles produce unusual antimicrobial compounds that could lead to new antibiotics. The study of DNA repair in radioresistant bacteria has provided insights into cancer and aging, both of which involve DNA damage. Extremophile research has even influenced materials science. The molecular structures that stabilize proteins at high temperatures or extreme pH have inspired the design of synthetic materials with enhanced stability. Biofilms from extremophiles have suggested new approaches to protective coatings. The economic and scientific value of extremophiles has led to concerns about their conservation and the ethics of their exploitation. Some unique extreme environments have been damaged by sampling or development. International efforts are underway to establish guidelines for responsible extremophile research and to protect extreme environments as biological reservoirs. Part 5. Origins of Life, Chapter 21. Rethinking the origin of life, perhaps the most profound impact of the hydrothermal vent discovery has been on theories about the origin of life itself. For decades, scientists debated where and how life first appeared on Earth. The discovery of chemosynthetic ecosystems at vents opened up new possibilities and challenged existing theories. The classical view, popularized in the 1950s by the Miller-Urey experiment, held that life originated in a primordial soup, a warm, shallow pond or ocean where organic molecules accumulated and spontaneously organized into living systems. In this scenario, lightning or UV radiation provided energy to drive chemical reactions in a reducing atmosphere, producing amino acids, sugars, and other building blocks of life. This theory had appeal. It was testable, and the Miller-Urey experiment demonstrated that amino acids could indeed form under plausible early Earth conditions. But it had problems too. The early Earth was probably not as reducing as Miller and Urey assumed. The primordial soup would have been dilute, making it difficult for organic molecules to concentrate enough to interact. And the warm, shallow environment would have been exposed to UV radiation that could destroy complex molecules as fast as they formed. The discovery of hydrothermal vents suggested an alternative. Life might have originated at vents, where chemical energy was abundant, organic molecules could be concentrated by thermal and chemical gradients, and the environment was protected from surface radiation. This idea gained support from several observations. First, the chemical conditions at vents are conducive to the synthesis of organic molecules. The hot, mineral-rich vent water contains hydrogen, methane, ammonia, and other reduced compounds. When this water mixes with seawater, chemical reactions occur that can produce amino acids and other organic molecules. Second, the mineral surfaces at vents could serve as catalysts, accelerating chemical reactions and perhaps serving as templates for the organization of organic molecules. The sulfide minerals deposited at vents have surface properties that could facilitate the polymerization of amino acids into proteins or nucleotides into RNA. Third, the energy gradients at vents, thermal gradients, chemical gradients, pH gradients, could drive the formation of complex molecules and the emergence of metabolic-like chemical cycles. Some researchers have proposed that the first metabolism was not biological but geochemical, occurring spontaneously in vent systems. Fourth, the environment at vents is relatively stable and protected. Unlike surface environments, which experience wide temperature swings, intense radiation, and asteroid impacts, the deep ocean provides a buffered environment where prebiotic chemistry could proceed for millions of years. These ideas coalesced into what's known as the hydrothermal vent origin of life hypothesis. Proposed by various researchers in the 1980s and 1990s, this hypothesis suggests that life began at or near hydrothermal vents, possibly at a specific type of vent called alkaline vents, which we'll discuss shortly. Chapter 22. The RNA World and Early Metabolism. To understand how life might have originated at vents, we need to consider what the first living systems might have looked like. Modern cells are enormously complex, with DNA storing genetic information. RNA transferring that information, and proteins carrying out most cellular functions. This interdependence poses a chicken and egg problem. DNA requires proteins to replicate, proteins require RNA to be synthesized, and RNA requires proteins for its synthesis. How could such a system arise spontaneously? One influential answer is the RNA world hypothesis. This proposes that early life was based on RNA, not DNA, and proteins. RNA is unique among biological molecules in that it can both store genetic information, like DNA, and catalyze chemical reactions, like proteins. Some RNA molecules, called ribozymes, can even catalyze their own replication. If RNA molecules could both store information and carry out metabolism, then a self-replicating RNA-based system could emerge without requiring the full complexity of modern cells. Over time, this RNA world could evolve proteins, which are more efficient catalysts than ribozymes, and DNA, which is more stable for long-term information storage. The hydrothermal vent environment could have facilitated the emergence of an RNA world. The mineral surfaces at vents could concentrate nucleotides and promote their polymerization into RNA. The temperature gradients could drive cycles of melting and annealing that would allow RNA to replicate. And the chemical energy available at vents could power early metabolic reactions. Some researchers have gone further, proposing that the first metabolic pathways mirror the chemistry occurring spontaneously at vents. The reverse citric acid cycle, a metabolic pathway used by some modern bacteria to fix CO2, has chemical steps that can occur non-biologically under hydrothermal vent conditions. Perhaps this pathway emerged first as geochemistry, then became biochemistry as organisms evolved to catalyze and control the reactions. This idea, that metabolism preceded genetics, is controversial but intriguing. It suggests that the first organisms might not have had genes at all, but were instead self-organizing chemical systems maintained by the flow of energy through the vent environment. Only later did these systems evolve genetic machinery to stabilize and propagate themselves. Chapter 23: Alkaline Vents and the Origin of Cells. While black smokers and other high-temperature vents have captured public attention, scientists studying the origin of life have focused on a different type of vent: alkaline hydrothermal vents. Alkaline vents are cooler than black smokers, typically 40 to 90 degrees Celsius, and emit fluids that are alkaline rather than acidic. The most famous example is the Lost City vent field, discovered in 2000 on the Mid-Atlantic Ridge. Lost City is not a typical mid-ocean ridge vent. It's located off-axis, and the vents are driven not by volcanic heat, but by serpentinization, a chemical reaction between seawater and mantle rocks that produces heat, hydrogen gas, and alkaline fluids. The Lost City vents form tall, white carbonate chimneys, some over 60 feet high, with a delicate, Gothic spire-like structure. The vents have been active for at least 30,000 years and possibly much longer, far exceeding the typical lifespan of black smoker vents. Why are alkaline vents relevant to the origin of life? Several reasons. First, the chemistry is milder than at black smokers. The moderate temperatures and near-neutral to alkaline pH are more compatible with the stability of organic molecules. Second, alkaline vents create natural pH gradients. The vent fluid is alkaline, pH 9 to 11, while the surrounding Hadean Ocean was likely mildly acidic due to dissolved CO2. This pH gradient across vent structures could have driven the synthesis of ADP, the energy currency of cells, even before the evolution of biological machinery. Third, the porous structures of alkaline vent chimneys could serve as natural compartments, concentrating organic molecules and creating protocell-like environments. The mineral walls could act as scaffolds for early membranes and as catalysts for chemical reactions. Fourth, serpentinization produces hydrogen gas, which can reduce CO2 to organic molecules. This process, still used by some modern archaea, might represent one of the most ancient metabolisms. A scientist named Nick Lane has championed the alkaline vent hypothesis, arguing that the natural proton gradients at these vents mirror the proton gradients that modern cells use to generate ADP. He proposes that early cells evolved not to create these gradients, but to harness gradients that existed naturally in the vent environment. According to this idea, the first cells were essentially molecular parasites on geochemistry. They didn't need to pump protons to create a gradient. The vent did that for them. They only needed to evolve membrane proteins that could harness the gradient to synthesize ADP. Later, as organisms moved away from vents, they evolved the machinery to create their own gradients. This is a radical proposal, but it has the advantage of explaining why all modern cells, bacteria, archaea, and eukaryotes, use the same basic mechanism, proton gradients, to generate ADP. It's a universal feature of life, suggesting it arose very early, perhaps because it was built into the environment where life originated. Chapter 24. Evidence and debates. The hydrothermal vent origin of life hypothesis is compelling, but is it correct? Scientific debates continue, and researchers are working to test the hypothesis through experiments and observations. One line of evidence comes from laboratory experiments simulating vent conditions. Researchers have shown that amino acids, sugars, and even more complex organic molecules can form under conditions similar to those at alkaline vents. Mineral surfaces from vents can catalyze the polymerization of nucleotides into RNA-like molecules. Lipid vesicles, cell-like structures, can form and remain stable under vent conditions. These experiments demonstrate that key prebiotic chemistry can occur at vents. But demonstrating that something can happen is not the same as proving it did happen. Many of the same reactions can also occur in other environments: hydrothermal pools on land, ice-covered oceans, even in space on comets and asteroids. Another line of evidence comes from the universal tree of life. By comparing the genes of all living organisms, scientists can work backward to infer the properties of the last universal common ancestor, Luca, the organism from which all modern life descended. Studies suggest Luca was a thermophilic organism that lived in a chemically rich, oxygen-free environment and used hydrogen and CO2 in its metabolism. This description fits a vent organism. While it doesn't prove Luca lived at a vent, it's consistent with that possibility. However, skeptics raise several objections. First, the vent environment is harsh. High temperatures and extreme chemistry could destroy complex organic molecules as fast as they form. While alkaline vents are milder than black smokers, they're still challenging environments for delicate prebiotic chemistry. Second, vents are ephemeral. They turn on and off over timescales of decades to centuries. Could life originate in such an unstable environment? Proponents counter that the vent system as a whole, spread across the global mid-ocean ridge, is stable even if individual vents are not. Third, some researchers argue that alternative environments are more plausible. Hydrothermal pools on land, with cycles of wet and dry conditions, could concentrate organic molecules more effectively than vents. Subsurface environments and porous rocks could provide stable conditions for prebiotic chemistry. Even the interface between ocean and atmosphere has been proposed as a likely site for life's origin. The debate remains unresolved. What's clear is that the discovery of chemosynthetic ecosystems at vents expanded our understanding of where life can exist, and therefore where it might have originated. Whether or not vents were the actual site of life's emergence, they've fundamentally shaped how we think about the origin of life. Chapter 25. What the vents teach us about early life. Regardless of whether life originated at hydrothermal vents, the vent ecosystems provide valuable insights into the conditions and challenges faced by early life on Earth. The early Earth, over 3.5 billion years ago, was a very different place than today. The atmosphere contained little to no oxygen, the sun was about 30% dimmer, and the planet was bombarded by asteroids and comets. Surface environments were likely harsh and unstable, but the deep ocean would have been relatively buffered from these disturbances. Hydrothermal systems would have existed from very early in Earth's history, as soon as there were oceans and volcanic activity. These systems would have provided stable chemical energy even as surface conditions fluctuated. The organisms living at modern vents are adapted to conditions that may resemble those of early Earth: oxygen-free or low-oxygen environments, chemically driven energy systems, high temperatures, and exposure to metals and sulfides. In this sense, vent ecosystems might be analogous to ancient ecosystems, not identical, but similar in their basic structure and function. Some of the metabolisms found at vents are thought to be very ancient. Methanogenesis, sulfate reduction, and hydrogen oxidation are all metabolic pathways that could have operated on early Earth and are still used by vent organisms today. Studying these organisms provides a window into ancient biochemistry. Moreover, the vent ecosystems demonstrate that life can persist and thrive without photosynthesis, an important lesson for understanding early Earth. Photosynthesis did not evolve until later in Earth's history. First billion years or more, life was entirely chemosynthetic and likely confined to environments like vents, where chemical energy was available. The vent ecosystems also demonstrate the importance of symbiosis in evolution. The partnerships between chemosynthetic bacteria and vent animals are examples of how different organisms can cooperate to exploit resources neither could access alone. Similar partnerships may have been crucial in the early evolution of eukaryotic cells, which arose from symbioses between different types of microorganisms. Finally, the extremophiles at vents, particularly the hyperthermophiles, provide clues about the nature of early life. Some researchers have proposed that the last universal common ancestor, Luca, was a thermophile, adapted to high temperatures. The fact that many deeply branching lineages in both bacteria and archaea are thermophiles supports this idea. However, others argue that this pattern doesn't necessarily mean Luca was a thermophile. It might mean that thermophiles are better at surviving mass extinctions and therefore are more likely to leave descendants. The debate continues, but vent organisms are central to these discussions. Part 6. Astrobiology Implications Chapter 26. Life Beyond Earth. The new possibilities. The discovery of life at hydrothermal vents transformed astrobiology, the study of life in the universe. Before 1977, the search for extraterrestrial life focused almost exclusively on planets with conditions similar to Earth's surface: moderate temperatures, liquid water, and sunlight. The possibility of life in our solar system seemed limited to Mars and perhaps Venus. The vent ecosystems changed this paradigm completely. If life can thrive in total darkness, at extreme pressures and temperatures, powered by chemical energy rather than sunlight, then potentially habitable environments become far more common, not just on planets, but on moons, in subsurface oceans, and perhaps even in interstellar space. Suddenly, the icy moons of the outer solar system, Europa, Enceladus, Titan, became prime targets in the search for life. These moons are covered in ice and receive little sunlight, but they might harbor subsurface oceans with hydrothermal vents. If Earth's vents can support life, why not vents on Europa? Even Mars, long considered a marginal candidate for life because of its cold, dry surface, gained renewed attention. The Martian surface may be inhospitable, but what about deep underground, where volcanic heat might melt ice and create hydrothermal systems? The discovery of extremophiles also expanded the types of biosignatures, signs of life, that astrobiologists look for. Instead of focusing solely on oxygen or other photosynthetic byproducts, scientists began considering the chemical signatures of chemosynthetic life: methane production, sulfide oxidation, unusual isotopic ratios in carbon and sulfur. Let's examine some specific targets in the search for life, all of them inspired or informed by what we've learned from hydrothermal vents. Chapter 27. Europa. Ocean world. Europa, one of Jupiter's four largest moons, is slightly smaller than Earth's moon and covered entirely in a thick shell of ice. For decades, scientists have known that beneath this ice lies a global ocean of liquid water, an ocean that may contain twice as much water as all of Earth's oceans combined. How do we know this ocean exists? Several lines of evidence. First, Europa's surface shows features consistent with a floating ice shell, cracks, ridges, and chaotic terrain that suggest the ice is moving and breaking up over a liquid substrate. Second, magnetic field measurements from NASA's Galileo spacecraft indicated the presence of an electrically conducting layer beneath the ice, consistent with a salty ocean. Third, The Hubble Space Telescope has detected water vapor plumes erupting from Europa's surface, suggesting that water from the subsurface ocean occasionally breaches the ice shell. But does this ocean harbor life? That depends on whether it provides the three key requirements. Liquid water? Yes. Chemical building blocks? Probably. And energy? Maybe. The energy question is crucial. Without sunlight, Europa's ocean would need another energy source. The most likely source is tidal heating. As Europa orbits Jupiter, the giant planet's gravity flexes the moon's interior, generating heat through friction. This heat could maintain the ocean in liquid form and might drive hydrothermal activity on Europa's seafloor. If Europa has hydrothermal vents, if hot water rich in minerals and reduced chemicals rises from the rocky seafloor into the overlying ocean, then it could host ecosystems similar to Earth's vent ecosystems. Chemosynthetic bacteria could use the chemical energy from the vents, and more complex organisms might evolve to exploit this productivity. This is not science fiction. It's a testable hypothesis based on what we know about Europa's structure and what we've learned from Earth's vents. The question is, how do we test it? NASA and ESA are planning missions to Europa. NASA's Europa Clipper, scheduled to launch in the 2020s, will make dozens of flybys of Europa, studying its ice shell, ocean, and chemistry. It will search for active plumes and analyze their composition. If organic molecules or chemical signatures of biological activity are detected in these plumes, it would provide strong evidence for life. A future mission might attempt to land on Europa and drill through the ice to sample the ocean directly. A formidable engineering challenge, but one that would provide definitive answers about whether life exists in Europa's ocean. The discovery of life at Earth's hydrothermal vents makes the possibility of life in Europa's ocean seem plausible rather than fanciful. We know that life can exist without sunlight, that it can thrive in cold, dark, high-pressure environments, and that chemical energy can support productive ecosystems. Europa checks all these boxes. Chapter 28. Enceladus. The plume world Enceladus, a small moon of Saturn, is even more intriguing than Europa in some ways. At only 300 miles in diameter, Enceladus is tiny. You could fit it inside Texas. Yet this little moon has proven to be one of the most exciting targets in the search for extraterrestrial life. In 2005, NASA's Cassini spacecraft detected massive plumes of water vapor and ice particles erupting from cracks near Enceladus's south pole. These plumes shoot hundreds of miles into space, creating one of Saturn's rings, the E-ring, which consists largely of ice particles from Enceladus. Subsequent observations revealed that Enceladus, like Europa, harbors a global subsurface ocean. The plumes provide a direct sample of this ocean, conveniently delivered into space, where spacecraft can analyze them without needing to land or drill. Cassini flew through these plumes multiple times, analyzing their composition. The results were astonishing. The plumes contain not just water ice, but also salt, suggesting a salty ocean in contact with rock, simple organic molecules including methane and molecular hydrogen. The presence of hydrogen is particularly significant. Hydrogen gas is produced when hot water reacts with rock in a process called serpentinization, the same process that drives alkaline vents like Lost City on Earth. The detection of hydrogen in Enceladus's plumes strongly suggests that hydrothermal activity is occurring on the moon's seafloor. Moreover, the ratio of hydrogen to methane in the plumes is out of equilibrium. There's more hydrogen than would be expected if only geochemistry were at work. On Earth, certain microorganisms consume hydrogen and produce methane. Could the same be happening on Enceladus? This is speculative, but it's based on solid observations. Enceladus has a subsurface ocean, contact between water and rock, hydrothermal activity producing hydrogen, organic molecules, an energy source, tidal heating from Saturn. All the ingredients for chemosynthetic life are present. The challenge now is to confirm whether life actually exists. Cassini was not equipped to detect life. It was a general-purpose planetary science mission. A future mission to Enceladus would need instruments specifically designed to detect biosignatures, complex organic molecules, unusual isotopic ratios, patterns that suggest biological activity. Such a mission would not need to land on Enceladus or drill through the ice. It could simply fly through the plumes repeatedly, collecting and analyzing samples. This makes Enceladus one of the most accessible targets in the search for life beyond Earth. Chapter 29. Mars. The subsurface frontier. Mars is often portrayed as a failed Earth, a planet that once had liquid water and a thicker atmosphere, but lost them billions of years ago, becoming the cold, dry desert we see today. The search for life on Mars has focused largely on the ancient past, looking for fossils or chemical signatures of organisms that might have lived when Mars was more hospitable. But the discovery of extremophiles on Earth, particularly those living deep underground, has opened a new possibility. Perhaps life still exists on Mars, not on the surface but in the subsurface. Earth hosts a vast, deep biosphere, microbial ecosystems living in rocks and sediments kilometers below the surface. These organisms survive on chemical energy from rock-water interactions, often in environments isolated from the surface for millions of years. If such ecosystems exist on Earth, why not on Mars? Mars's interior still contains heat from its formation and from radioactive decay. This heat could maintain liquid water in deep aquifers or hydrothermal systems. In such environments, Martian microorganisms could persist independent of surface conditions. Evidence for subsurface liquid water on Mars has grown stronger in recent years. Radar observations suggest the presence of liquid water beneath the south polar ice cap. Seasonal features called recurring slope lineae it might be caused by briny water seeping from underground. Methane has been detected in Mars's atmosphere, and its concentration varies seasonally. This methane could be produced by geological processes, but biological production remains a possibility. If life exists in Mars's subsurface, it would almost certainly be microbial and chemosynthetic, similar to Earth's deep biosphere organisms. Detecting such life would be extraordinarily challenging. Surface rovers cannot drill deep enough to reach the subsurface, and even if they could, distinguishing Martian life from terrestrial contamination would be difficult. Future missions might deploy seismometers to detect subsurface liquid water, drill rigs to access deep aquifers, or instruments to analyze the chemistry of gases escaping from underground. The search for subsurface life on Mars is a long-term endeavor, but one informed by what we've learned from Earth's extremophiles. Chapter 30. Exoplanets and the habitable zone beyond our solar system. Astronomers have discovered thousands of exoplanets, planets orbiting other stars. Many of these planets are potentially habitable, and the search for life has focused on rocky planets in the habitable zone, the region around a star where temperatures allow liquid water to exist on a planet's surface. But the discovery of chemosynthetic ecosystems challenges the concept of the habitable zone. If life doesn't require sunlight, then the habitable zone becomes much broader, or perhaps the concept becomes less useful. Consider a rocky planet or moon with a subsurface ocean, heated by tidal forces or radioactive decay. Such a world could harbor life even if it's far from its star, receiving little to no sunlight. The planet might be in the habitable zone in a traditional sense, but life could exist in its subsurface regardless. This realization has led astrobiologists to expand their criteria for habitability. Instead of focusing solely on surface conditions and stellar radiation, they now consider the presence of liquid water, surface or subsurface, available chemical energy, photosynthetic or chemosynthetic, geophysical activity, plate tectonics, volcanism, tidal heating, atmospheric composition for surface life, planetary protection from radiation. Planets that might have been dismissed as uninhabitable because they're too far from their star or tidally locked or have thin atmospheres might actually harbor subsurface life sustained by chemosynthesis. This doesn't mean we should ignore the traditional habitable zone. Planets with surface water and photosynthesis remain the easiest targets for detecting life because photosynthetic organisms produce atmospheric oxygen, a strong biosignature. But it does mean we should keep an open mind about other possibilities. The discovery of life at Earth's hydrothermal vents has made the universe seem much more hospitable than we once thought. Wherever there is liquid water and a source of chemical energy, life might find a way. Chapter 31. Biosignatures and Detection Strategies. Finding life beyond Earth requires knowing what to look for. What biosignatures would indicate the presence of life? The discovery of chemosynthetic ecosystems has expanded the catalog of potential biosignatures. For photosynthetic life, atmospheric oxygen is a strong biosignature. Oxygen is highly reactive and doesn't accumulate in atmospheres without continuous production by photosynthetic organisms. Detecting oxygen, or its byproduct, ozone, in an exoplanet's atmosphere would be compelling evidence for life. But chemosynthetic life doesn't produce oxygen. What biosignatures might it produce? Methane is one possibility. Methanogenic organisms produce methane as a waste product. On Earth, most atmospheric methane is biological, produced by bacteria in wetlands, rice paddies, and the guts of animals. On another world, significant atmospheric methane, especially if found with water vapor and carbon dioxide, could indicate biological activity. However, methane can also be produced geologically through serpentinization or volcanic activity. Distinguishing biological from geological methane requires additional evidence. Isotopic ratios, seasonal variations, or the presence of other gases that correlate with biological activity. Hydrogen is another gas of interest. While hydrogen itself is not a definitive biosignature, it's produced by various geological processes. An unusually low concentration of hydrogen in an environment where it should be abundant might indicate consumption by microorganisms. Complex organic molecules could also serve as biosignatures. If spectroscopy detects molecules like amino acids, lipids, or nucleotides in an exoplanet's atmosphere or ocean, this would suggest biological activity. Life tends to produce organic molecules with specific structural features, chirality, molecular handedness, specific size distributions, and patterns that differ from abiotic synthesis. Unusual isotopic ratios can also indicate biological activity. Life preferentially uses lighter isotopes of elements like carbon and sulfur, creating distinctive patterns that can be detected spectroscopically. On Earth, carbon isotope ratios have been used to identify ancient microbial life in rocks billions of years old. For subsurface oceans like those on Europa or Enceladus, biosignatures might include elevated concentrations of metabolic byproducts, methane, hydrogen sulfide, etc. In plumes, specific ratios of gases out of chemical equilibrium, complex organic molecules of biological origin, patterns in molecular structures, e.g. even-numbered carbon chains and lipids. Isotopic signatures inconsistent with purely geological processes. Detecting these biosignatures requires sophisticated instruments. Future telescopes, like the James Webb Space Telescope and proposed missions like LUVOIR or HABEX, will analyze exoplanet atmospheres with unprecedented precision. Missions to Europa and Enceladus will carry mass spectrometers and other instruments designed to detect biological molecules. But perhaps the greatest challenge is interpretation. How do we distinguish between biological and geological signals? How confident must we be before claiming detection of extraterrestrial life? The scientific community is developing frameworks for evaluating biosignature evidence, recognizing that the claim of extraterrestrial life will require extraordinary evidence. Chapter 32. The search continues. The search for life beyond Earth is entering an exciting new phase. Informed by decades of studying extremophiles and chemosynthetic ecosystems, scientists are now planning missions that have realistic prospects of detecting life if it exists. In our solar system, the priority targets are Europa, with its subsurface ocean and probable hydrothermal activity, NASA's Europa Clipper and ESA's JUICE, Jupiter Icy Moons Explorer. Missions will characterize Europa's ocean and search for signs of habitability. A future lander or submarine mission could search for life directly. Enceladus, where active plumes provide samples of the subsurface ocean. A dedicated Enceladus mission could fly through the plumes repeatedly, analyzing their chemistry for biosignatures. This is arguably the most accessible, potentially habitable environment beyond Earth. Mars's subsurface, where liquid water may exist and chemosynthetic life might persist. Future missions might include deep drilling or seismic networks to detect subsurface aquifers. Titan, Saturn's largest moon, which has surface lakes of liquid methane and a subsurface water ocean. While very different from Earth, Titan might host exotic forms of life adapted to its chemistry. NASA's Dragonfly mission will explore Titan in the 2030s. Beyond our solar system, the search focuses on exoplanets. Thousands have been discovered, and characterization is improving. Future telescopes will analyze exoplanet atmospheres for biosignatures, oxygen, methane, unusual chemical combinations, or other signs of life. The discovery of extremophiles has made scientists optimistic but cautious. We know life is adaptable and can thrive in unexpected places, but we also know that detecting life is difficult. Even on Earth, with samples in hand, distinguishing biological from geological processes can be challenging. Doing so with remote observations across interplanetary or interstellar distances will be even harder. Yet the search continues, guided by what we've learned from hydrothermal vents and extremophiles. If life arose on Earth in extreme environments, it might have done the same elsewhere. And if we find it, even if it's just microorganisms in Europa's ocean or Enceladus's plumes, it will be one of the most profound discoveries in human history. It will mean we are not alone. It will mean life is robust, adaptable, and perhaps common in the universe. And it will all trace back to that day in 1977 when scientists first saw life thriving in boiling ocean water. Part 7. Ongoing Research and Future Directions, Chapter 33. Returning to the vents, the study of hydrothermal vents did not end with the initial discoveries. On the contrary, it accelerated. Over the past 4 decades, scientists have returned to vent fields repeatedly, conducting ever more sophisticated observations and experiments. Modern vent research uses technologies that were unavailable in 1977. Autonomous underwater vehicles, AUVs, can map vast areas of seafloor without human pilots. Remote-operated vehicles, ROVs, equipped with high-definition cameras, manipulator arms, and sampling equipment can work at vents for hours. Some sites have been instrumented with permanent observatories, providing continuous data on vent chemistry, temperature, and biology. One of the most ambitious projects was the installation of cabled observatories at several vent sites. These observatories include sensors, cameras, and experimental platforms connected to shore by submarine cables. They provide real-time data on vent conditions 24 hours a day, year-round, something impossible with periodic submersible visits. These observatories have revealed the dynamic nature of vent ecosystems. Vents don't just flow steadily. They pulse, fluctuate, and sometimes shut off entirely. Earthquakes can trigger changes in vent flow. Volcanic eruptions can bury vent communities under fresh lava. Then new vents open and new communities establish. Observatories have documented these events in real time, showing how organisms respond to changes in their environment. When a vent suddenly increases its flow, do organisms migrate? When a vent shuts off, how long does the community persist? These questions are now being answered through long-term observations. Scientists have also conducted experiments at vents, using submersibles to deploy instruments and collect time series samples. They've measured the growth rates of tube worms, the metabolic rates of vent bacteria, and the chemistry of mixing between vent water and seawater. They've studied how larvae find vents, how communities recover after disturbances, and how different species interact. Genomic techniques have revolutionized vent biology. Scientists can now sequence the genomes of vent organisms, revealing the genes responsible for their extreme adaptations. They can measure gene expression in real time, showing how organisms respond at the molecular level to changes in temperature, chemistry, or pressure. Metagenomic studies, analyzing the DNA of entire communities, have revealed vast microbial diversity at vents. For every species of bacteria cultured in the lab, dozens more exist that we cannot yet culture. These uncultured bacteria might have novel metabolisms, unique adaptations, and potentially valuable enzymes. Chapter 34. New Discoveries. New mysteries despite decades of study. Hydrothermal vents continue to surprise scientists. New vents discovered in recent years have revealed ecosystems unlike anything seen before. In 2000, the discovery of the Lost City vent field introduced alkaline vents to the world. These vents, as discussed earlier, are driven by serpentinization rather than direct volcanic heat. They form towering carbonate structures and support ecosystems dominated by microbial communities rather than large animals. Lost City has become a key site for studying the origin of life and ancient metabolisms. In 2012, scientists exploring the Cayman Trough in the Caribbean discovered the world's deepest known vents at nearly 16,000 feet. The extreme pressure at this depth creates unique conditions, and the vent fauna includes species found nowhere else. Vents have been discovered in unexpected locations. In the Arctic under permanent ice cover, in shallow waters just a few hundred feet deep, and even in lakes. Lake Baikal in Siberia has cold seeps with chemosynthetic communities, showing that such ecosystems are not limited to the ocean. One of the strangest discoveries came from the Antarctic, where scientists found vents beneath the ice shelf. These vents support communities of vent organisms in total darkness, beneath ice, in one of the most remote and inhospitable regions on Earth. Each new vent field reveals new organisms. Scientists have now described hundreds of vent species, but estimates suggest thousands more remain undiscovered. The global diversity of vent fauna is still being cataloged, and new expeditions regularly discover new species. Some of these discoveries have challenged existing theories. For instance, scientists found vent communities in sediment-covered areas where no one expected vents to occur. They discovered mobile organisms, fish and crustaceans, that migrate between vent fields, something previously thought impossible given the isolation of vent communities. Perhaps most intriguingly, scientists have discovered vent organisms living not at vents, but in other environments. Some vent bacteria have been found in whale falls and cold seeps, as expected, but others have been found in deep-sea sediments far from any obvious chemical energy source. How are they surviving? Are they dormant, waiting for conditions to change? Or have they found ways to exploit trace chemical energy in sediments? These mysteries keep scientists returning to the vents, deploying new technologies, and asking new questions. Chapter 35. Threats to vent ecosystems. Hydrothermal vent ecosystems, once thought to be beyond human reach, now face threats from human activities. The deep ocean is no longer as remote as it once was, and vents have become targets for exploitation. Deep-sea mining is the most immediate threat. The mineral-rich deposits at hydrothermal vents, massive sulfides containing copper, zinc, gold, and other metals, are economically valuable. Companies have obtained exploration licenses for several vent sites, and commercial mining could begin within years. Mining would devastate vent ecosystems. The process would involve removing the mineral chimneys and surrounding rock, the very habitat where vent organisms live. Even if mining avoided active vents, the sediment plumes generated by mining operations would spread for miles, potentially smothering nearby ecosystems. The ecological consequences could be severe. Vent organisms are highly specialized and slow to recover. If a vent field is destroyed by mining, recolonization could take decades or centuries. Some species with limited ranges might be driven to extinction. Scientists and conservationists have called for a moratorium on deep-sea mining until its impacts are better understood. They argue that we are on the verge of destroying ecosystems we barely understand, potentially losing species with unique adaptations before we've even discovered them. Climate change poses another threat. While vent ecosystems are largely isolated from surface conditions, they are not entirely immune. Changes in ocean chemistry, particularly ocean acidification, could affect vent organisms. Rising temperatures could alter ocean circulation patterns, potentially affecting the dispersal of vent larvae. Pollution is also a concern. Even the deep ocean is not pristine. Microplastics have been found at the deepest ocean trenches, and chemical pollutants have been detected in deep-sea organisms. While vents are far from major sources of pollution, they are not immune. Tourism represents a potential future threat. As technology improves and costs decrease, deep-sea tourism might become feasible. Visits by tourist submersibles could disturb fragile vent ecosystems or introduce contamination. Finally, there's the threat of scientific research itself. Collecting samples, deploying instruments, and conducting experiments all have impacts on vent ecosystems. While scientists try to minimize these impacts, the accumulation of decades of research might have affected some well-studied vent sites. Balancing scientific study, conservation, and potential economic exploitation of vents will be one of the challenges of the coming decades. International agreements on the protection of vent ecosystems are being negotiated, but progress is slow. Chapter 36: Conservation and Management. Recognizing the threats to vent ecosystems, scientists and policymakers have begun developing frameworks for their conservation and sustainable management. Some vent fields have been designated as Marine Protected Areas, MPAs. For instance, the Endeavour hydrothermal vents on the Juan de Fuca Ridge were designated as Canada's first MPA in 2003. The designation prohibits mining and other destructive activities while allowing scientific research. International bodies like the International Seabed Authority, ISA, are developing regulations for deep-sea mining that include provisions for protecting vent ecosystems. The challenge is balancing economic interests with conservation. Mining companies argue that regulations are too restrictive, while conservationists argue they're too lax. One proposal is to establish a network of protected vent sites, ensuring that representative examples of different vent types and their associated ecosystems are preserved. This would allow mining at some sites while guaranteeing that others remain untouched for scientific study and conservation. Scientists are also working to establish baseline data on vent ecosystems. Comprehensive surveys of species diversity, abundance, and ecosystem function at key sites. These baselines are essential for detecting changes and assessing the impacts of future activities. Genetic banks are being established to preserve the genetic diversity of vent organisms. Tissue samples and cultured bacteria are stored in repositories, ensuring that even if populations are lost, their genetic material remains available for study and potential restoration. Education and outreach are also important. Most people are unaware that vent ecosystems exist, let alone that they face threats. Scientists are working to raise public awareness about these remarkable ecosystems and the need to protect them. International cooperation is essential. Hydrothermal vents occur in international waters and within the exclusive economic zones of many nations. Effective conservation requires agreements that span national boundaries and involve all stakeholders. The coming decades will be critical. Decisions made now about the use and protection of vent ecosystems will determine whether future generations can study and marvel at these extraordinary habitats or whether we'll lose them before fully understanding their importance. Chapter 37: Technology and the Future of Vent Science. The future of hydrothermal vent research will be shaped by advancing technology. New tools and techniques are enabling observations and experiments that were impossible just years ago. Autonomous underwater vehicles, AUVs, are becoming more sophisticated, capable of mapping large areas of seafloor with high-resolution sonar and cameras. Some AUVs can operate for weeks at a time, covering hundreds of miles and documenting vent fields across entire ridge segments. These vehicles are also becoming smarter. Using machine learning algorithms, they can identify interesting features, like venting fluids or unusual organisms, and adjust their surveys accordingly. In the future, AUVs might autonomously discover new vent fields and conduct initial characterizations before human scientists even know they exist. ROVs, remotely operated vehicles, continue to improve as well. Modern ROVs can work at depths exceeding 20,000 feet, carry sophisticated sampling equipment, and remain on the seafloor for days. Some are equipped with manipulator arms precise enough to extract individual bacteria from biofilms or collect single-tube worm larvae. New sensor technologies are providing unprecedented detail about vent chemistry and biology. In situ mass spectrometers can analyze the chemistry of vent fluids in real time, detecting dissolved gases, and organic molecules. Genetic sequencers can identify organisms on the seafloor, providing species identifications without bringing samples to the surface. Imaging technology is also advancing. High-definition and 3D cameras provide detailed views of vent communities. Some systems use fluorescence or other techniques to visualize features invisible to normal cameras, like distributions of bacteria or the flow of water through sediments. Environmental DNA, EDNA, sampling is revolutionizing how scientists study vent ecosystems. By collecting water samples and analyzing the DNA they contain, researchers can detect organisms without seeing or capturing them. This technique can identify rare species, track larvae, and reveal the presence of microorganisms too small or cryptic to observe directly. Genomic and transcriptomic techniques allow scientists to study not just what organisms are present, but what they're doing. By analyzing gene expression, researchers can determine which metabolic pathways are active, how organisms are responding to environmental conditions, and how they interact with symbionts. Laboratory simulations of vent conditions are also improving. High-pressure aquaria and flow-through systems can maintain vent organisms in conditions similar to their natural habitats. These systems allow controlled experiments on vent organisms, something impossible in the deep ocean. Some scientists are even proposing human-occupied facilities at vent sites, underwater research stations where scientists could live and work for extended periods. While technologically challenging, such stations would enable observations and experiments impossible with brief submersible visits. Chapter 38. Unresolved questions. Despite decades of intensive study, many fundamental questions about hydrothermal vents remain unanswered. These questions drive ongoing research and will likely occupy scientists for decades to come. One major question concerns the evolution of vent fauna. How did these highly specialized organisms originate? Did they evolve from shallow water ancestors that adapted to progressively deeper and more extreme conditions? Or did they evolve in the deep ocean, perhaps in ancient vent systems? Genetic studies have provided some answers, showing that vent tube worms are related to other polychaete worms and that vent clams are related to shallow water clams. But the timing and sequence of evolutionary transitions remain unclear. When did the symbioses between animals and chemosynthetic bacteria evolve? Did they evolve once and then spread to different lineages, or did they evolve independently multiple times? Another question concerns the global biogeography of vent organisms. We know that different ridge systems host different species, but we don't fully understand the patterns or mechanisms of dispersal. How do larvae travel between vent fields separated by hundreds or thousands of miles? What barriers limit dispersal? How do new vents get colonized? The role of vents in global ocean chemistry is also not fully understood. Vents inject enormous quantities of heat, metals, and chemicals into the ocean. They influence the chemistry of seawater, the cycling of elements like iron and sulfur, and potentially the ocean's carbon cycle. But quantifying these effects requires knowing how many vents exist globally and how their output varies over time. Information we don't yet have. The microbial diversity of vents remains largely unexplored. Culture-based techniques can isolate only a small fraction of vent bacteria. Metagenomic studies reveal vast diversity, but most species remain uncharacterized. What metabolisms do these organisms use? What ecological roles do they play? What symbiotic relationships exist beyond the well-studied tube worm and clam systems? The impacts of disturbances on vent ecosystems are another active area of research. Volcanic eruptions can bury vent communities under fresh lava. How quickly do they recover? What succession patterns occur as ecosystems rebuild? Do the same species return, or does the community composition change? Finally, there's the question of how vent ecosystems will respond to future changes, whether from climate change, ocean acidification, or direct human impacts. Are vent ecosystems resilient to disturbance, or are they fragile and slow to recover? These questions ensure that hydrothermal vent research will remain vibrant and active for the foreseeable future. Chapter 39. Interdisciplinary connections. The study of hydrothermal vents has become increasingly interdisciplinary, connecting fields that rarely interact. This convergence of different scientific perspectives has led to new insights and unexpected discoveries. Geology and biology have been closely linked since the beginning of vent research. Understanding the geological processes that create vents, plate tectonics, volcanism, hydrothermal circulation, is essential for understanding the ecosystems they support. Geologists and biologists now work together routinely, combining geological surveys with biological sampling. Chemistry has always been central to vent research, but the development of new analytical techniques has deepened the integration. Geochemists study the chemistry of vent fluids, biogeochemists examine how organisms alter chemistry, and biochemists investigate the molecular adaptations that allow organisms to survive. Physics plays an important role as well. Understanding fluid dynamics, how hot vent water mixes with cold seawater, is crucial for understanding vent ecosystems. Physicists also contribute to understanding heat flow, pressure effects, and the physical properties of extreme environments. Microbiology has become increasingly important as scientists have recognized the central role of microorganisms in vent ecosystems. The symbionts in tube worms and clams are just the beginning. Vents harbor extraordinary microbial diversity, much of it still unknown. Genomics and molecular biology have revolutionized vent research, allowing scientists to study the genetic basis of extreme adaptations, the evolutionary relationships among vent organisms, and the molecular mechanisms of symbiosis. Computer science and engineering contribute through the development of autonomous vehicles, sensors, and data analysis tools. Machine learning algorithms help identify patterns in the vast datasets generated by vent research. Astrobiology has become intertwined with vent research, as the vent ecosystems provide models for how life might exist on other worlds. Astrobiologists study vent organisms to understand the requirements for life and the biosignatures it might produce. Even the social sciences have connections. Anthropologists and sociologists study how human communities interact with the ocean, including how decisions about resource use and conservation are made. Economists analyze the costs and benefits of deep-sea mining versus conservation. This interdisciplinary nature is one of the strengths of vent research. By bringing together diverse perspectives and expertise, scientists can address questions too complex for any single discipline to tackle alone. Chapter 40. The legacy of discovery. The discovery of life at hydrothermal vents represents more than a scientific advance. It's a shift in how we understand life and our planet. Before 1977, we thought we knew the rules. Life requires sunlight, moderate temperatures, and specific chemical conditions. We thought the deep ocean was a marginal environment where life barely persisted. We thought Earth's ecosystems all traced their energy back to the sun. The vent ecosystems showed us that life is more diverse, more adaptable, and more creative than we imagined. They revealed that entire ecosystems could exist on chemical energy alone. They demonstrated that organisms could thrive in conditions we thought were lethal. This discovery has had ripple effects across science. It changed biology, forcing a re-evaluation of life's boundaries and capabilities. It changed ecology, revealing new types of ecosystems and food webs. It changed geology, showing the interconnections between geological processes and life. It changed astrobiology, expanding the search for life beyond traditional habitable zones. The discovery also changed how we think about our planet. Earth is not just a surface phenomenon with a few organisms living in the depths. The deep biosphere, organisms living in rocks, sediments, and hydrothermal systems deep underground and under the ocean, may constitute a significant fraction of Earth's biomass. We live on a planet where life extends from the upper atmosphere to miles beneath the surface, exploiting every available energy source. This expanded view of Earth's biosphere has implications for how we manage our planet. If vast ecosystems exist in the deep ocean, they deserve consideration in our resource use decisions. If microorganisms in the deep subsurface play important roles in global element cycles, we need to understand these systems before we disrupt them through mining, drilling, or waste disposal. The legacy of the 1977 discovery extends beyond science to culture and philosophy. The vent ecosystems capture the imagination, alien-looking organisms thriving in extreme conditions inspire wonder and curiosity. They remind us that Earth still holds mysteries, that exploration and discovery are ongoing, that there are still frontiers to explore. For the scientists who made the initial discovery and those who followed, the vents have been a source of endless fascination. 45 years later, every dive still brings surprises. Every new vent field reveals something unexpected. Every technical advance enables new observations. This is the nature of scientific discovery at its best. One breakthrough leads to new questions, new technologies, new insights. The 1977 discovery opened a door, and scientists have been walking through it ever since, discovering a world stranger and more wonderful than anyone imagined. Conclusion Epilogue. The continuing journey on February 17th, 1977, when Alvin descended to the Galapagos Rift and discovered life thriving in superheated water around hydrothermal vents. The scientists aboard did not know they were witnessing one of the most important biological discoveries of the century. They were looking for evidence of hydrothermal activity, warm water venting from the seafloor. What they found changed everything. The giant tube worms, massive clams, dense bacterial mats, and swarming crustaceans challenged every assumption about what life requires and where it can exist. The subsequent realization that these ecosystems were powered by chemosynthesis, not photosynthesis, overthrew a fundamental paradigm of biology. For the first time, we knew that life could exist independent of the sun. The implications rippled through science. Biologists had to reconsider the definition of an ecosystem and the requirements for life. Microbiologists discovered vast new groups of organisms, thermophiles, hyperthermophiles, and chemosynthetic symbionts. The recognition of archaea as a third domain of life fundamentally reorganized the tree of life. Geologists gained new insights into ocean chemistry and the role of hydrothermal circulation in planetary processes. Astrobiologists expanded their search for life beyond Earth, recognizing that potentially habitable environments might include ice-covered ocean moons, subsurface aquifers on Mars, and perhaps environments we haven't even imagined yet. The discovery continues to influence science today. Every year brings new findings from vent ecosystems, new species, new metabolisms, new insights into evolution and adaptation. The development of extremophile enzymes for industrial use has created entire biotechnology industries. The study of vent communities informs our understanding of early Earth and the origin of life. But perhaps most importantly, the vent discovery reminds us of the power of exploration. The scientists aboard Alvin in 1977 were not looking for a revolutionary discovery. They were conducting routine geological research. The presence of abundant life was completely unexpected. Yet they had the wisdom to recognize that what they were seeing was extraordinary and the humility to admit that their previous understanding was incomplete. This is the nature of science. Remain open to evidence, even when it contradicts expectations. Be willing to revise theories when observations demand it. Recognize that nature is often stranger and more complex than our models suggest. The story of hydrothermal vents also reminds us how much remains unknown about our own planet. The deep ocean covers more than 70% of Earth's surface, yet we've explored only a tiny fraction of it. How many more vent fields remain undiscovered? How many species are still unknown? What other ecosystems might exist that we haven't even imagined? These questions drive ongoing research. New technologies, autonomous vehicles, advanced sensors, genomic tools are enabling discoveries impossible just years ago. International collaborations are mapping the global distribution of vents and documenting their ecosystems. Long-term observatories are revealing the dynamics of vent communities over time. At the same time, these ecosystems face new threats. Deep-sea mining could destroy vent habitats before we fully understand them. Climate change may alter ocean conditions in ways that affect even the isolated deep sea. The challenge is to balance scientific exploration, conservation, and sustainable use, to learn from these ecosystems while protecting them for the future. The journey that began in 1977 continues. Each new expedition brings discoveries. Each new technology enables observations previously impossible. Each new species documented adds to our understanding of life's diversity and capabilities. Where will this journey lead? Perhaps to the discovery of life on Europa or Enceladus, in subsurface oceans heated by tidal forces and supporting chemosynthetic ecosystems like Earth's vents. Perhaps to new understandings of how life originated on Earth and what forms it might take elsewhere. Perhaps to biotechnologies we can't yet imagine, based on the molecular adaptations of extremophiles. What is certain is that the 1977 discovery of life at hydrothermal vents fundamentally changed how we view life and our place in the universe. It showed us that life is more resilient, more adaptable, and more persistent than we thought. It revealed that our planet harbors extraordinary ecosystems in its most extreme environments. It opened our minds to the possibility that we might not be alone in the universe. The discovery reminds us that Earth is still a place of mystery and wonder, that exploration and discovery are ongoing endeavors, and that nature still has the power to surprise us. Even in the 21st century, with all our advanced technology and scientific knowledge, we can descend into the ocean depths and find things we never expected. Ecosystems that rewrite the textbooks and force us to reconsider what we think we know. This is the legacy of the hydrothermal vent discovery, a reminder that the universe is vast and full of wonders, that life is extraordinarily adaptable, and that the next great discovery might be waiting just beyond the edge of our current knowledge, in the darkness of the deep ocean or in the icy oceans of distant moons. The scientists aboard Alvin in 1977 looked out into the darkness and saw something impossible, life thriving where it should not exist. Their discovery changed science forever. And the story continues, with each new dive, each new observation, each new revelation about the remarkable adaptability of life. In the end, the discovery of life in boiling ocean water taught us the most important lesson: expect the unexpected. Nature is more creative than our theories, more diverse than our models, and more surprising than our predictions. The universe is full of possibilities we haven't yet imagined, and that is why we continue to explore. Closing, this has been the story of one of the most profound scientific discoveries of the 20th century, the discovery of chemosynthetic ecosystems at hydrothermal vents. From the initial descent of Alvin to the Galapagos Rift in 1977, through decades of research revealing the extraordinary adaptations of extremophiles to the current search for life on other worlds, this discovery has transformed our understanding of life and its possibilities. We've explored the historical context that led to the discovery, the remarkable biology of vent organisms and their bacterial symbionts, the geochemistry that powers these ecosystems, and the implications for the origin of life on Earth and the search for life beyond. The journey from that first glimpse of giant tube worms in Alvin's lights to our current understanding has been one of continuous revelation. Each answer has led to new questions. Each discovery has opened new avenues of research. The story is far from over. Scientists continue to explore hydrothermal vents, discovering new species, documenting ecosystem dynamics, and unraveling the molecular mechanisms that allow life to thrive in extreme conditions. Future missions to Europa, Enceladus, and other ocean worlds may reveal whether the lessons learned from Earth's vents apply to life beyond our planet. As we look to the future, the 1977 discovery reminds us that the universe is full of wonders waiting to be discovered, that life is more adaptable than we imagine, and that exploration, whether of the deep ocean or deep space, continues to reveal profound truths about nature and our place within it. The scientists who found life in boiling ocean water opened a door to a new understanding of our planet and the cosmos. We are still walking through that door, and the journey ahead promises to be as remarkable as the journey so far.